only be activated by the burst firing. So those are generally my summaries. And then, like, if you're interested more, this result is published in eNeuro, and then also the molecular level work done by Mary and me is just published in JPCB. And that's pretty much it for the first part. Okay. So we get back to our initial dopaminergic signaling model. So we have that system, we got the dopamine release, etc. But this time we got psychostimulants. We would like to see how it will get changed if we have some psychostimulant molecules inside the simulation. And what are those psychostimulants? Basically, I mean, well, all of us drink coffee every day, and then like the coffee is the main psychostimulant, but it's not that addictive. So, you know, we're not interested in this. But the other two that I'm talking about is the cocaine, and the other one is the amphetamine, are two of the, let's say, well, addiction-making, the addictive psychostimulants. And like we would like to see in a spatial context how they will affect the dopamine metabolism, the dopamine neurotransmission. And basically, the cocaine has been well studied for a long time. And then, like, the main mechanism is the competitive inhibition. It binds the dopamine transporter in the outward facing, and it will just close the place that the dopamine passed through. The dopamine is reuptaken, and then like it will be out of the picture for a long time. And when we are doing our simulations, we are just simulating our system for 10 seconds, and the half-life of the cocaine is going up to hours. So basically, we need to define a molecule that diffuses through, binds to the dopamine transporter, and stops its function. Which, I mean, like the basic modeling strategy for this, the cocaine is that. And then when we implemented this, what we are doing is we use the cocaine has a higher binding affinity compared to the dopamine molecules. It's actually 10 times higher. So it did a huge effect. And if you look at the results, I tried two different concentrations of the dopamine, cocaine. And one of them is the IC50 value. The other one is the half of the IC50 value. I just want to see how this concentration is going to affect. And then again, in a similar setting with the previous results with the different dopamine transport clusters, if you look at the histograms, I see that the IC50 level on the IC50 level cocaine will increase the dopamine concentrate, the extracellular dopamine concentrations with significant amounts. Because like if you compare with the green and the magenta line, it is way higher at the higher concentrations. And on the other hand, the half IC50 value did not have a huge effect. And I would like to understand why this effect is not that high. And then the main reason is because of the equilibrium dynamics. Because if you remember from the previous time, a lot of inward phasing dopamine transporters. And when your outward phasing dopamine transporters are started to get occupied by the cocaine molecules, some of them is going back to the outward phasing state to bring back to the homeostasis, and then it will just start clearing up more dopamine molecules. So that's an interesting finding. Of course, there are a lot of other areas that can be investigated with the cocaine. This is the study that I did right now. And I'm also interested in the amphetamine, and then my colleague from Bahar Lab, Dr. Big Liu, created a model of amphetamine-induced dopamine efflux model. And this model is looking quite complicated. There are many interactions in this. But let me make some of the interactions clear. And one of them is the amphetamine transport through the dopamine transporter. Amphetamine will get inside the cell. It's not like, in the cocaine view, it was easy because the effect is only on the extracellular side. There's nothing happening at the intracellular. But for this time, it goes to the cell, and then it will start interacting with the other molecules. And the two of the main interactions that the amphetamine process is that One of them is the phosphorylation of the dopamine transporter, and then as a result of this phosphorylation, dopamine started to release, dopamine transporters started to release dopamine molecules to the extracellular space, and so it will induce the, it will interact with the Rho, and then like the Rho protein, and then Rho protein will induce the endocytosis of the dopamine transporters. And well, it's a complex model. There are 49 equations and 66 parameters, but more than that, if you try to put all of them in a spatial context, like a real M-cell model, you will end up with billion molecules, and it's impossible to see how it's going to happen. So basically, we would like to come up with a simplified version of this model. And the way I did this is basically this is our normal model. Normal means like the original model that I described in the previous part. And then we add similarly an amphetamine intake, amphetamine uptake pathway, and in addition to that, this amphetamine calls the phosphorylation of the outward phasing dopamine transporter to the phosphorylated outward phasing version, and this amphetamine also calls the endocytosis of this outward phasing dopamine transporter. And of course, the phosphorylated one has an efflux. It will get the dopamine from the interior of the cell, it will bind it, and then it will start releasing dopamine molecules to outside. And yeah, basically this is just those two mechanisms. And what we basically did, there are a lot of diffusion events needs to happen for this phosphorylation or this endocytosis event to happen. Because the molecule will, like amphetamine will, let's say, interact with Rho will go find the GTP, Rho-GTP complex will go and cause endocytosis. But modeling all these diffusion events will be so hard. So basically what we did is we put everything on amphetamine, and we assume that all the diffusion is modeled as amphetamine diffusion, and the rest of the events is going to happen. We need to find out a way how the amphetamine concentration is related with the rate of endocytosis and with rate of that phosphorylation. And Bing had a bunch of simulations with different amphetamine concentrations. And I used that information, and I fit amphetamine concentration to the phosphorylation rate and the endocytosis rate. And then I basically every half a second simulation stops, calculate number of amphetamine inside the cell, get the concentration of that, and then use that information to feed your M-cell model. What is your rate constants for the cytosis, for the phosphorylation of the dopamine transporter? And then after every half a second cycle, it is going on, and then that will be just like an online update on the rate of the amphetamine induced endocytosis and phosphorylation in a spatial setting, because we are adding the diffusion of the amphetamine to a reaction model. And basically what we find out is we tried this in two different, uniform, I don't know, it's not realistic, but we just want to see the limits of the model. And when we put it to uniform setting, what we find out is it's increasing the extracellular dopamine concentration with a very, very large number. And then we would like to understand why this is, because it is reuptake of the amphetamine is so high in this case. From the previous part, I mentioned that this uniform distribution of the dopamine transporters has more effective clearance of the dopamine, right? In the same way, there will be more amphetamine molecules inside, and then if you have more amphetamine molecules, it will cause more efflux, more endocytosis, and you will end up with more dopamine molecules. On the other hand, if you go to the non-uniform, which was our case, which does not have that efficient clearance of the dopamine in the previous part, and it does not have an efficient clearance of the amphetamine also. So it's not taking amphetamine inside the cell, and then the efflux rate is so low. So as a result, our normal case and then all two amphetamine concentration cases have pretty much the same result. And very, very low concentrations, of course, the effects are not that obvious, but when we change the the dopamine transporter distribution, we see a big difference. I mean, the competitive inhibition mechanism for cocaine does not have a significant effect at the low concentration, and the effect of the amphetamine is gone when because of the low uptake of the amphetamine under non-uniform distribution. And the concentration, actually, well, I forgot to mention that the effect of the concentration is very minor under non-uniform distribution. It's because of the low reuptake rates, and this is like this part. In the last part, I talked about the dopaminergic signaling, but we're changing some gears, and then I'm going to talk about the glutamatergic signaling. And again, in the same synapse picture, but our transporters and our receptors and everything is changed. And similarly, we will get an action potential, and then these voltage-gated channels are start to take some calcium, and calcium induces the vesicular release of the glutamate molecules. But instead of dopamine receptors, it will go to these IGLURs, generally namely AMPA receptors and the NMDA receptors. It will activate them, start taking some ions inside the postsynaptic cell, and in the postsynaptic cells, it will activate the molecule calcium calmodulin dependent kinase type 2. That molecule is a very central piece for memory and learning. And yeah, I just want to talk about like what is CAMK2 and why it is important. CAMK2 is one of the main drivers of the synaptic plasticity, and it is very highly expressed compared to other kinases. It's like 1 or 2% of the protein in your brain, and it is connected to many signaling molecules. And more it's a very structurally complex molecule. It's a dodecamer with 2 rings, and then there are like multiple phosphorylation sites and binding sites for each subunit, and then there are 12 of them, so it becomes a very hairball-like network structure. And so I just mentioned what is synaptic plasticity. I just want to define that it's the strengthening or the weakening of the synapses over time depending on the activity patterns. For example, you will have, for example, one type of it is the long-term potentiation. You will have high-frequency potentiation input for a short period of time, but the activity will stay very long time. And it's and it is related to memory and learning, of course. And the version, which is the long-term depression, which has low-frequency input for a long time, which will lead to a low activity level for a very long time after the stimulation is gone. But for the CAMK2 is generally related to long-term potentiation, and I just want to talk to you about like what is the role of CAMK2 in the synaptic plasticity. So basically, CAMK2, similar to like in the long-term potentiation, after its activation from calcium calmodulin complex, it will stay active for a long time. And the active CAMK2 will phosphorylate these AMPA receptors, which is one type of the IGLURs that I mentioned, and then it will it will phosphorylate them, and as a result of this phosphorylation, it will have a higher conductivity, and it will start taking more ions inside the postsynaptic cell, and it will bring more activity. And also, it induces the recruitment of additional AMPA receptors to the brain, so that like there will be more ions transported inside the cell, and then it will do more. And CAMK2 has multiple states. I mentioned that it's a very, very interesting molecule. And then, like, the main thing is, like, there are 3 different components of the CAMK2. And one of them is the calmodulin binding, 2 distinct phosphorylation sites in different locations. Basically, it has an association domain, it's a kinase domain, and then there's a linker between them. And on the linker, there are 2 different phosphorylation regions. One of them is the T288. 86, the other one is the T305 and 306. They're so close to each other, so I just, for the sake of modeling, I'm just using them as one single. And basically, in the beginning, it's if there's no activity, it's generally in the DUU state, which is the no calmodulin, no phosphorylation on both of the phosphorylation sites. Upon calmodulin binding, it will go into the CUU state, which has calmodulin on it, but there's no phosphorylation, but it starts getting active. And then the most interesting mechanism is this, because an active CAMK2 can phosphorylate its neighbor, this. And then like there are more mechanism to that. But what we are